Chapter 16, the citric acid cycle. Okay, remember we're going to be over the next few chapters, starting here with chapter 16, we're going to be discussing cellular respiration. Okay, cellular respiration is where pyruvate is taken and it is consumed. It is further oxidized to yield water and carbon dioxide. That whole reason you have to breathe out. So when we go and we look at this citric acid cycle, you've been taught since you were a kid, and you started taking biology courses, junior high, high school, whenever, glycolysis, glucose to pyruvate into the citric acid cycle, and so on and so forth. And I've been trying to get it across to you over the past few weeks in lecture that it's not just pyruvate that feeds into this. Just like with glycolysis, it wasn't always just glucose that feeds into that process. So, everything you eat, you consume, has the potential to be shuttled into the citric acid cycle. The amino acids, fatty acids, take the fatty acids, triglycerides, phospholipids, whatever, cut every two, boom, boom, boom. Add a CoA, now you have acetyl-CoA. Glucose to pyruvate, remove one of the three carbons of pyruvate, you now have the 2 carbon acetyl, add that to CoA. Amino acids, go through, remove the amino, do some, cuts and cleaving, and here you go. You now have 2 carbons, acetyl-CoA. And the big thing about the citric acid cycle is not the ATPs that are going to be made due to substrate level phosphorylation. It is the redox reactions. The harvesting of all of these excited electrons, which will be transferred over to NADH and FADH2, and then carried to the electron transport chain. Notice also here in this artist's rendition of the citric acid cycle, it's a cycle. There's no beginning, there's no end. Things will feed out, things feed into this constant mishmash of enzymatic reactions. So it's the respiratory chain, the electron transfer. That's what we ultimately are going to be using to harness all of this energy right there in those excited electrons. This is what's going to drive the majority of the ATP that our cells are then going to be able to use. So, let's talk what it takes to get this acetyl-CoA. Now, if you look here, this is coenzyme A. This starts with a base nucleotide, adenine, add on phosphate, carbon 3. The carbon 5 has dual phosphates, and then this pantothenic acid, long carbon chain, tipped by beta-mercaptoethylamine. Wow, that one's a nice fun one to say. But notice right here, this thiol group, this sulfur-containing functional group right there. This is where our acetyl group, 2 carbons, acetyl. This is where we're going to get this carbon-sulfur bond, which will end up giving us acetyl-CoA. Now you'll notice here this shorthand, instead of always writing out coenzyme A, which is what this entire thing is, they write CoA and then .sh, referencing this thiol group. Now to get the pyruvate in, Pyruvate just can't clip and attach to coenzyme A. Pyruvate's in the cytoplasm. The CoA is actually in the mitochondria. So first you're going to have a Symporter. Okay, Symporter is a type of a transporter that usually couples the movement of hydrogen through the transporter from extremely high to extremely low concentrations, just basically going down, concentration gradient. Symporter couples the movement of the hydrogen with something else. Here, this carrier couples the movement of hydrogen with the transport of pyruvate across the outer membrane. It's going to come into contact, then the pyruvate's going to come into contact with what is referred to as Pyruvate Dehydrogenase complex, PDH for short, Pyruvate Dehydrogenase. This is a cluster of complexes of enzymes and proteins. Why am I being so so non-specific? Because there's a lot of proteins here, a lot of enzymes, multiple copies of the same proteins, the same enzymes that are all going to come together. So we're starting off here. 1, 2, 3. 3 carbons. Sorry, had to throw in a joke there. So our first step, oxidative decarboxylation. Oxidative, we're going to be doing an oxidative reduction here. We're going to be removing a hydrogen. And we're going to decarboxylate, meaning we're going to lose a carbon. Coenzyme A is going to work with our PDH complex. The E1, E2, E3 is just showing the enzymes involved in the pyruvate dehydrogenase complex. Here are some accessories that are needed to be there, TPP, lipoate, and FAD. NAD is going to be reduced to NADH. And in the process, the first carbon will be cleaved and released. Where's this hydrogen, this electron coming from? Remember the thiol group here, SH, that was the terminal end of the coenzyme A. Well, I got to lose the hydrogen there so that now you can have the bond from carbon to sulfur. So there it goes. You look right here, the delta G, negative 33.4. This is a very exothermic process. I'm not saying exothermic reaction because 1, 2, 3, 3 reactions all at once to go from pyruvate to acetyl-CoA. So the overall process is extremely exergonic. Our enzymes that we're looking at here, pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. Our coenzymes, our coplayers, thiamine pyrophosphate, TPP, lipoate coenzyme A, okay, FAD and NAD. The lipoate is coenzyme, has 2 of these thiol groups, these 2 sulfur-containing functional groups that have a tendency to go cyclic, oxidized form, reduced form. What this means is that it acts as the hydrogen carrier. The hydrogen that was coming from the coenzyme A will be held here. Boom. It'll then be transferred on to the NAD when the acetyl group replaces it. So you end up with an acetylated form, a reduced form, oxidized form. Boom, 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 boom. It bounces between these. What's interesting is this is not a free enzyme. This is an enzyme that is covalently attached to enzyme 2, the dihydrolipoyl transacetylase. When you look at it, this right here is a colorized space filling model. No, not a space filling model. Topographical micrograph, I guess is the best way to say it. They went through, used a scanning electron microscope and whatnot to take pictures of this entire complex. And they went through and colorized it so you can tell the differences between the proteins. So you have this entire, looks like a flowering plant, rosetta. E1, E2, E3, here's the lipoate. So E2, our dihydrolipoyl transacetylase, is the core. These leaf-like extensions you see here, that's the lipoate. That's the ones we were just talking about on the last slide. Pyruvate dehydrogenase, 1, 2, 3. And then 1, 2, 3 are these clusters here of dihydrolipoyl dehydrogenases. Notice the colors are different there, colors different there. That means that there's multiple proteins, multiple coenzymes that are associated there to form this complete working structure. It's also to denote that there's multiples. It's not just E1, just one E1 protein is all of that. There could be multiple of them here all clustered together, because you look here at the core. The E2 core can have numerous copies of dihydrolypyl transacetylase. It's not just the 4 that this figure is trying to show. These are multiples of them that are all clustered together, complex together, and then you got all these complexes forming one giant cluster. We're going to see that overall shape, the way those things are arranged, what proteins, what coenzymes are there, is going to be a theme in multiple steps throughout the citric acid cycle. Alpha-ketoglutarate dehydrogenase, and then there's an alpha-keto acid dehydrogenase that's going to come into play later. Enzymes 1 and 2 may be some variability. Enzyme 3 is nearly identical in all of these complexes. So they're looking at this as that all of these clusters and all these proteins and whatnot are paralogs of each other. Means that way, way back, a common ancestor had gene duplication occur. It happens. Fast forward, thousands of generations, millions of years or whatever, and those duplicated genes accumulated different mutations, different adaptations, selected for different mutations across the different copies. And well, one of those copies was PDH, the next one was, KDH, in the next one, and so on and so forth. When we look at PDH, what we notice again, you have these clusters of enzymes and proteins, each doing different jobs. Pyruvate dehydrogenase, okay. First thing it does is cleave carbon 1, right there, CO2. Reason you have to breathe out. TPP acts as a carrier for this 2-carbon thing, because remember, 2 carbons is pretty small. It can easily get lost in the mix, so you have to have something here that's going to take it and move it to the next dihydrolipoyl transetylase. Okay, it is then picked up by lipolysine, becomes acetyl-lipolysine. It's transferred here to another, transferred to another, transferred to another, and then it's released, Acetyl-Coa. So at no point in time here are these two carbons ever released. They go from pyruvate, held, now carried along by coenzyme A. The two excited hydrogen electrons, known as the hydrogens, get passed off to FADH2, which then passes them on to NADH, which goes on to, the electron transport chain. So when you go through step 1, decarboxylation, step 2, oxidation, you notice that at no point in time, like I said, are these released. Esterification, electron transfer to regenerate, and then you take off, regenerate the FAD by reducing NAD to NADH. They refer to this continuous passing on as substrate channeling. This is different than many of the other metabolic pathways we've seen, where an enzyme takes its substrate, turns it into product, releases it, and then the next enzyme in that pathway will take the product, which is now its substrate, turns it into a product, releases it. So at no point in time is there a direct interaction between the enzymes. That's different here because you have a carrier that's taking it from enzyme 1 and immediately moving it over, transferring it to something else, which now is going to go to enzyme 2. So at no point in time do those two carbons ever released. This substrate channeling is more efficient. It's faster. It's more efficient, it's faster, less waste, and takes less energy. That's what this tethers intermediates means. No point in time do those two carbons ever released. They are always held and then move from one to the next, to the next, to the next by some protein or part of the protein complex.